Hello everyone, welcome to our third segment of our virtual field trip called Science in the Corridor. My name is Sarah Gomes and I'm a graduate student working on my master's in geography. I study the green hair-streaked butterfly, and much of the work that I do is in the green hair-streaked corridor. So today I'll be sharing with you a little bit about my research, as well as some other ways that science is happening in the corridor and ways that you can get involved too. So if you're following this virtual field trip in order, you may remember from parts 1 and 2 that there are 11 restored habitat sites within the green hair-streaked corridor. Now, of those 11 sites, the green hair-streaked butterflies mainly inhabit 3 of those sites. That's Quintara Steps, Rocky Outcrop, and Grandview Park, all 3 of which you've seen in part 2. I'm really curious as to why that is, and this is one of the big picture questions that really guides my research. You may have also noticed in part 2 that each of those sites seems a little bit different than the other. Some sites seem a little windier, some sites seem a little foggier, and this is because each site has its own microclimate that's influenced by the topography and the built environment around them. I'm really interested in how these microclimates differ, and more specifically, how coastal fog impacts habitat quality for these butterflies. Of those 3 sites that I mentioned, all 3 of which are on the western hillside. This means that they face the Pacific coast, and they're some of the first sites to intercept that really dense coastal fog that comes in from the Pacific Ocean. Because of this, I suspect that coastal fog may have some sort of long-term legacy effect on overall plant function that makes these habitats more attractive to the green hair-streak than some of the others. My thesis research consists of 2 overarching components, the first of which is a controlled fog experiment, and the second is a set of butterfly observations in the field. For the controlled fog experiment, I focus on the seaside daisy plant, which you may remember from part 2 is one of the favorite nectar plants of the green hair-streak. In this experiment, I place the seaside daisies inside the chambers behind me, and then I measure their response with the portable photosynthesis system. Essentially, what this allows me to do is take the heartbeat of a plant. When we go to the doctor, they take our heartbeat with a stethoscope, and that tells them how blood is flowing through our body. With this machine, I basically do the same thing. I clamp onto a leaf, and it tells me in real time how water and gases are flowing in and out of the plant. As I mentioned, I place the seaside daisies inside of one of 2 chambers. This here is the fog chamber, and behind it is the control chamber. And they're constructed exactly the same, but what sets them apart is an ultrasonic humidifier, also known as a fog machine. I plug the hose into the machine, the water comes in, it simulates fog, and pumps it up through this tubing into the plastic chamber, and this allows me to simulate a natural fog event. On the inside of each chamber, I have several instruments. We have installed weather stations, soil moisture probes, and leaf wetness sensors. And all of these instruments allow me to track and manipulate the microclimate on the inside of these chambers. This allows me to control exactly how much fog and how much irrigation each plant receives, and then I measure their response with the portable photosynthesis system. To understand plant response, I focus on photosynthesis rates. And what I've learned through this experiment is that fog will enhance photosynthesis rates regardless of soil water content. Here in San Francisco, we live in a semi-arid Mediterranean ecosystem. And this means that often years we don't get a lot of rain, and soil moisture will remain dry for most of the year. We know that coastal fog can help alleviate some of that drought stress through 2 mechanisms. The first of which is through fog drift directly to the soil. Many of the plants in foggy environments have evolved over the years to capture some of this fog water and funnel it down to the root system to support photosynthesis. mechanism, some plants can actually take the water in through the leaves, and this is what we refer to as foliar uptake. In this experiment, we've also learned that seaside daisies are well adapted to these foggy environments and that they'll rely on foliar uptake methods when soil moisture is low. So in other words, during drought years like this year, coastal fog becomes even more important to help plants survive. My research project is still in progress, so one of the next steps that I'll be taking is to connect the dots between what I've learned about plant function to how this actually impacts butterflies in the field. To do this, I'll be surveying the amount of nectar rewards available to adult butterflies and pairing that with butterfly observations in the field. I'll also be exploring some of the other reasons why butterflies might choose some sites over others, and I'll be comparing things like plant composition, microclimate differences, and other habitat suitability characteristics. I wanted to end this segment by spending some time talking about other ways that science is happening in the corridor and how you can get involved too. You may have heard Amber mention that we have 2 iNaturalist projects here. In case you're unfamiliar, iNaturalist is a really cool app created by the California Academy of Science that allows you to share your observations of the natural world with people from all over the world. How it works is you take a picture of your observation, you note the time and location, and when you upload it to iNaturalist, you share it with this broader scientific community. Oftentimes, when you upload an observation, it'll be included in citizen science projects near you. I've created 2 projects for the Green Hair Streak Corridor. The first is simply called the Green Hair Streak Corridor Project, and any observations seen in the Green Hair Streak Corridor will automatically be uploaded to this project. So far, we've seen over 850 species, we've had over 4,000 observations made by hundreds of people, and it's a really fantastic resource. Lastly, we have the Green Hair Streak Monitoring Project, which is very similar, but it singles out the coastal green hair streak butterfly. This allows me to see how green hair streak butterflies are moving throughout the corridor, and it helps Nature in the City evaluate the effectiveness of their restoration. Thank you all for watching today, and I encourage you to get out there, download iNaturalist, and make your own observations too.